Good afternoon, and thank you all for joining us for the keynote presentation of our Pursuit Award ceremony. I'm very pleased to announce this afternoon's keynote speaker, Dr. Amy Bastian. Dr. Bastian is a renowned neuroscientist and Chief Science Officer at the Kennedy Krieger Institute. She is also Director of the Motion Analysis Laboratory, where she studies neurocontrol of human movement. Dr. Bastian is a professor of neuroscience and neurology at Johns Hopkins University School of Medicine. She is a recognized and highly accomplished physical therapy research scientist whose interests include understanding cerebellar function and dysfunction, locomotor learning mechanisms, motor learning, and how to rehabilitate individuals with many types of neurological conditions. Today, Dr. Bastian will present her keynote titled Learning and Relearning Movement. We're really pleased to have Dr. Bastian with us this morning. Welcome.
Okay, thank you so much. So I'm honored to be here. I really enjoyed the morning, terrific talks, and I'm looking forward to meeting with students in particular. Students are my favorite, as well as the faculty. Of course, the generosity and insight of the Ward family, I think, has to be acknowledged. I'm particularly pleased to do this because it involves trainees, people who are starting their scientific career. And I really feel like that's where we should be putting our efforts, because this is the future, and you three were just fabulous. So thank you for coming all the way that you came, and I appreciate the generosity of the Ward family. Okay, everyone, what I'm going to do today is I am going to tell you pieces of different studies, and it's going to span more than a decade. So I'm not going to go into all the details on every experiment, but I'm going to try to tell you a story of how we approach movement control and why we've done the things that we've done. I'm going to focus most heavily on children, although we do study adults, and I will tell you a little bit about adult studies for reference. So it's no surprise to you that I would say movement is complicated, but let's talk about some numbers. You have 600 muscles in your body, a couple hundred bones, 80 joints in the arms and legs. Joints have different degrees of freedom, so they can rotate in different ways. And you also have, and I'm sorry to say this, slow, noisy sensors. Now what that means is that your vision, your proprioception, or your sense of limb position, somatosensory function, vestibular function, all these things don't actually work perfectly. They're noisy, so they're never exactly on target. And even more importantly, they're slow. And by slow, I mean that the time from which you sense something with your eyes, with your hands, with your legs, with your vestibular organs, it's about at least 100 milliseconds before it reaches the brain and it can actually influence what you do. And so I like to tell people, everybody here, everything you're seeing, everything you're feeling has already happened. And so that means that your brain, if it only uses feedback, is operating on outdated information. Boo. That's bad for movement control, because you want to be able to do amazing things like this, where you need to make good predictions about how your movement's going to unfold, so you don't have to rely on time-delayed feedback. And so today I'm going to tell you about a form of learning that we think is essential for developing these predictions about how your body's going to move in novel situations, and we're going to sort of hijack this mechanism to try to improve how we can rehabilitate people. So the thing I'm going to talk about is cerebellum-dependent learning. So for those of you who aren't neuroscience oriented, cerebellum means little brain. And it's this part of the brain down here. It's subcortical, so it's below the cerebral cortex. And this part of the brain seems to be an integral component of a specific kind of learning that I'm going to tell you about, and that we can drive in unusual ways. What I'll tell you today is that this kind of learning, and that is learning movement, changes not only how you move, but how you perceive your movements. I'm going to tell you about the fact that part of it develops during childhood, which I think is fascinating and has implications. And then I'll tell you that we've been able to use this to some extent to help people, children and adults, with damage to the cerebrum. And the cerebrum is the rest of the brain. So cerebral damage, of course, that's a gross oversimplification, but these are the kinds of folks that you see if they've had a stroke, or often tumors are in the cerebrum and other kinds of problems. So this is the outline of the talk that I'm going to give you today. And one thing that I want you to keep in mind is that we all know this. I love this picture. It's like a kid with a pacifier snowboarding. It's insane, right? And then this is my friend Joe. So children do appear to learn differently than adults. Most people would say they learn better. And what better is something that we should discuss. What I'll tell you is that better is not, it could be better, but it's also different. And I want to talk about some of the differences in the kind of learning that I'll tell you about today and elaborate further on that. Okay, so what we're going to do now is we're going to just dive right in. I'm going to tell you about a paradigm that we've used, and I'll tell you straight up, people have been using this kind of experiment for a long time. Started in cats. And actually, Janie Yang really has studied this in infants in amazing ways. And part of it, what I do, is motivated by her. She's at the University of Alberta, and one of her trainees who came to postdoc with me is here, Kristen Musselman. But we've really latched onto this as a learning paradigm. And so what I'm going to do is tell you how we can drive learning of new locomotor patterns using a treadmill that's split. So this treadmill, people can walk on, and as they walk on the treadmill, we record 3D motion capture from their body, which I'm not illustrating here, and we can measure all kinds of things, including the position of their feet, the joints of their legs, the joints of their arms and body. And so one of the measurements I'll tell you about, it's a very straightforward one, is how symmetric their stepping is. A step length here is the distance between the 2 feet when the heel touches the ground. So that would be one step, and that would be the other step length. And we can calculate the symmetry of this stepping, how even the steps are, as people walk on our treadmill, which will either move with the 2 belts tied, that is at the same speed, or split, where we make one belt go faster than the other one. Now you might ask, why on earth would you want to do that? And the answer is because it's fun. No, the answer is because it turns out that we can induce very interesting learning properties by having people exposed to this. And the thing I love about this paradigm is it's, we think, driven largely by subcortical structures. So that means you don't have to think about what you're doing. I don't have to tell you how to walk. I can put a 2-year-old on this, and if I can get them to watch TV, they'll still learn the new motor pattern. So this is a nice way of invoking lower motor centers in order to drive learning. Now, the basic paradigm that I'll be using for most of the talk today is illustrated below. This is time, and these are the speeds of the belts in meters per second. So 0.5 meters per second is a pretty slow saunter, and 1.5 is getting close to, a college student's maybe typical walking pace. We start our experiments with baseline trials where the belts are tied. They're moving at the same speed. And then at some point, we stop the treadmill and we start it back up and we split the belts. So one belt, in this case, is shown to go 3 times faster than the other one. After we expose people to this condition, they walk for 10 to 15 minutes, and then we go back to tied belts, where the belts, again, are moving at the same speed. The reason we do that is that allows us to see the effects of what we see being learned in the split belt condition. So let me show you what this looks like. Sometimes I'll show you animations in the form of what look like balloon men, and the reason I do this is because our adults do not like to be videotaped wearing spandex with markers all over their bodies. It's understandable. And so what we do is we animate this using custom software that we write in the lab. So what you're going to see are balloon guys walking along. So this first balloon guy is going to walk, and this is the tied belt baseline condition. So the belts are moving at the same speed, and you can see that the walking pattern, the steps are even. Every time the heel goes down, the steps are taken of the same size. What's happening below is a trace, a scrolling trace of the pendulum-like motion of the leg. And so when the leg is forward, it goes up. When the leg is back, it goes down. It's a vector from the hip to the toe. And what you can see here is the legs are oscillating around the same midpoint, right? Because this is symmetric, and they're a nice antiphase. Okay, now, if you're an adult or an older child and I expose you to this 3-to-1 split condition, you don't take multiple steps on one side for the other side. However, if you're really small, Kristen will tell you you do. By small, I mean 1. So let me show you what it looks like in a healthy adolescent. So this is the very first trial. Big step, small step, big step, small step. They look like they're limping. One leg is going 3 times faster. They take a big step and then a small step because one leg is getting yanked out from behind them on the fast belt. And you can see down here that the legs are no longer oscillating around the same midpoint. One leg is walking in front of the other one. And it's taking smaller, it's got a smaller stride, and they're taking smaller steps. So it's kind of like this. Does that make sense? Did you guys see it, or do you want me to do it over here? Okay, so you could walk like that all day long. You could do it. Some people do. But the nervous system does not want to do that. And so if you give people 10 or 15 minutes, this is the same split belt condition, they reestablish a normal pattern for these belts. So in other words, the legs are oscillating around the midline, they're in antiphase, and they're actually taking the same size steps, even though one leg is going 3 times faster than the other. Now you may think to yourself, how is that possible? Well, it has to do with where you put your feet and when you put your feet down on the treadmill. But suffice it to say that naturally, you do this all the time. And the reason you do this all the time is because you walk in curves. So if you walk in a circle, the outer leg has to take a bigger stride, the inner leg has to take a smaller stride. I didn't look like I was limping, right? That's because I've recentered how I'm taking my steps. So this is what is being learned here. There's more to it, and I'll tell you that later, but this is the phenomenon. Now, importantly, when we go back to tied belts, we see evidence of learning because people have to unlearn this calibration. They limp in the opposite direction, and they can't help it. They'll know it's coming, and they still do this. So this is exactly the same situation as the baseline. Okay, so this is the phenomenon. We're taking a movement you already know, we're exposing it to an unusual perturbation, we're looking to see how the nervous system learns it, and then we're looking to see if it gets stored in any way and you have to unlearn it. Okay, so if we measure this across time, I have a measurement that I'll call step asymmetry. If the asymmetry is 0, you're walking with nice even steps. If it's large or small in the negative or positive direction, that means there's an asymmetry. It's basically the difference between the 2 step sizes normalized to their sum. So here's the initiation of the split. So each of these dots represents a pair of steps, and you can see they're asymmetric. People learn. If I am tricky and I give them a little catch trial where I make the belts tied, they show me that they have an after effect, that they limp. I take them back to the split belt. And then once I return to regular treadmill tied belts, they actually have to unlearn it, and it's not immediate at all. It takes a good bit of time. You can see they're not back to baseline, to true baseline, until, a couple hundred steps. So this is not it's fast, but it's not that fast. So this is the process we're going to be talking about. And just to give you a spoiler alert, what I've shown you here is that in a healthy person, I can train in an after effect that gives them a limp. Well, what I'm going to show you later is that in somebody who limps, I can train in an after effect that makes them not limp. And I'm going to take advantage of that. But before I do that, I want to tell you about how this develops. And in order to tell you how this develops, I'm going to tell you about another phenomenon in adults that I think is important and that we're just starting to explore. So all I've shown you so far is a motor recalibration, but it turns out that this learning is also due to some changes in how you perceive your body to be moving. So I'm going to explain to you how we test this in order to make this point clear. At baseline, before we start the learning, what we do is we give people a simple psychophysical test. We put them on the treadmill. This is another balloon guy with really wide hips. And we ask them, we start their blue leg walking at a given speed. And then we ask them with the button box, they can't see their legs, to make their red leg match that speed. Does that make sense to everybody? It's really simple. So here's what it looks like. Blue leg starts. Okay, red leg is going to start. And they're hitting the buttons, the arrow buttons, until they try to make it match speeds, which is shown over here. I'm drawing where the belts are the same speed. Right here, there's the dotted line. This is time. And what you can see is this person's pretty good at this. So they have good proprioception. They're able to, or kinesthesia, you can label it what you want, but velocity perception of leg movement. And they do this nicely. So we do this 3 times to see how good you are. And then we do our usual adaptation, where we make you limp, you learn a new pattern, and then we go back to this. And this is the interesting part, because we also, in addition to changing the motor pattern, we change what feels good, what feels symmetric to you. So if I give this test right after learning, same thing, what you're going to see is that the red leg that had been trained fast now feels like it should go fast in order to feel even. So let me show you this. We start the blue leg, same speed we did before, and now the person is adjusting their red leg to make it feel exactly the same as their blue leg. Wow, yeah, isn't that cool? It's robust. So what you see is this person feels like the legs are moving at the same speed when the red leg is moving quite a lot faster than the blue leg. And what I've plotted as a function of time over here is how even the steps are. And what you'll notice is when they start, the steps are uneven, but actually they choose a speed that makes their steps nice and symmetric. So they're going back to their learned pattern, and they perceive that learned pattern to be even in speed, which reflects, that's not the truth, but that reflects their motor pattern is even. So this is pretty cool. So we can measure this. And if we measure this, what we do is we simply take the difference between these, gives us an idea of how much they've recalibrated their perception, and we do it all during unlearning, and then we can plot it as a function of time. So this is the initial time point across a group, and you can see that this perception decays. This would be normal perception, and this is perception that my red leg should be going faster in order to feel even. Now, an important point that's going to tie into what I'm going to show you with children is that they did not recalibrate their perception completely. So what that means is I gave them a 1 meter per second perturbation, the difference between 1.5 and 0.5. So this difference is 1. If they recalibrated their perception perfectly, it would go all the way up to 1 up here. But instead, it's only about 40%-ish. So this perception, we've tried lots of time courses, we've tried starting in different places. It doesn't explain, or it doesn't account for, you can think of it that way, the whole motor phenomenon. Instead, they only partially recalibrate their perception. Okay? So this suggests that there might be more than one learning process being layered on top of each other during this. And I'll explain that further in a minute, but just trust me when I say that this is the phenomenon that we see. Okay, now I'm going to take a little segue because I mentioned earlier that we like this paradigm because you use brain structures that are below the cerebral level, subcortical structures, to do this learning. And so I've highlighted the cerebellum here. And what I'm going to show you is the same kind of balloon guy trying to learn this task just by walking. And this is somebody who's had a cerebellar lesion. So they start out, we're going to have slower speeds, and you can see that their gait is what you might expect. They get uncoordinated. They have an irregular stepping pattern. When we split the belts, they can walk with the belts moving at different speeds. So they have the neural structures intact to be able to create a nice anti-phase walking movement. They don't fail entirely. That's important. We think that might be more spinal, but we don't have full proof. But you give them practice, and they don't seem to recalibrate. They don't optimize this. And then if you go back to tied belts, they don't store an after effect, so they don't limp in the opposite direction. So we've actually studied this in these patients a lot. And what it tells us is that they're unable to recalibrate their predictions of how their body's going to move on this treadmill. Now, that's not so good for them. And there's a whole nother line of my laboratory that works on that problem that I'm not going to tell you anything about today, unfortunately. But it might be good for others. Before I go on to talk about why it's good for others, let me just show you what a really significantly impaired individual looked like. They walk with symmetry at tied belts. At split, you don't see that nice learning curve. And then back at tied belts, you see minimal after effect. So this is like kind of the worst case scenario. Now, you might ask, well, do they change how they feel the belts? Do they change their perception of the belt speeds? Is that part of this or separate from this? And so when we study that, what we find is that they also have a deficit there. So what I'm showing you here is a different group of people with cerebellar damage and a matched control group. So purple is the controls, learning the split belt calibration. Here's that surprise catch trial we give. Here's a group of cerebellar subjects. They're not all as bad as the individual I just showed you. So they look like they learn a little bit. Their catch is smaller, so they don't store as much, and they never really get back to symmetry. Interestingly, if we test their perception of the belt speeds, what you see is that healthy people recalibrate their perception, like I showed you before, but the cerebellar patients do not. And so one possibility is that there's a piece of this learning that the cerebellar patients can take advantage of. It's not much, but it's a little. And then there's another piece of this that is perhaps cerebellum dependent, and that might be involved. This sensory recalibration might be a marker of that. So that's actually interesting to me because I'm interested in rehabilitating the cerebellar patients and taking advantage of whatever's left. But what's interesting for people with damage in other parts of the brain is using this cerebellum dependent mechanism. So before I go into that, do you have any questions? Okay, you should feel free to, I know it's been a long morning, so interrupt me at any given time. I'll try to tell some jokes to keep everybody awake. Now I'm going to talk about kids and how this develops. And so this is the background in adults. I've shown you a few things. Adults recalibrate their walking if we expose them to this predictable perturbation on a split belt treadmill. Adults recalibrate not only how they move, but how they feel they move. And if you damage the cerebellum, you get rid of part of that learning, and you also get rid of the calibration of how you feel you're moving. Okay. What do kids do? So this is my first Canadian postdoc. I'm going to highlight all of them later because for some reason motor control in Canada is fantastic, and you guys probably already know that. So I get imports, and most of them go back. Not all of them, but some of them have definitely chosen to leave the U.S., which we can discuss that later. So this is a study that Erin Vasudevan did. She was also a Janie Yang and a Paul Zerr trainee. And what we did is we just simply took 60 people and simply, haha. And we did all the kinematics, split belt treadmill learning, and this is what an adult group looks like. This is a really nice, tight adult group. There's the learning, you can hardly see the data points below it, and the unlearning of the split belt treadmill. Now, what I'm going to show you is how kids do on this. And this is interesting. This is the 3 to 5-year-old group. These are the ones that actually showed some learning. You get down to 3 and it's dicey for lots of reasons. 6 to 8-year-olds, 9 to 11-year-olds, 12 to 14, 15 to 17. You'll notice a couple things. The first thing you probably notice is that there's a lot more noise in the traces of the children. That's actually between subjects variance here. So this is, but if you look at an individual trial, trust me, they're still really variable. But the other thing you might notice is that these guys sort of look like they have a much more linear trajectory. It's not that nice sharp curve followed by a subtle curve, a double exponential. And only when you get older do you start to have that kind of nice double exponential. Okay, so the cerebellar patients that could learn kind of looked a little bit linear, if you remember. And so that's interesting. So let me show you another thing about children that I think is really important. We recently did a study where we decided to take a group that we know learns in this much more linear manner, and we compared them to adults doing the perceptual task that I just told you about to see if children also change not only what they do, how they move, but also how they feel they're moving. And so this is another group of 44 subjects, and the gray are adults, and the yellow are our 6 to 8-year-old children. And what you notice is, again, they're slow to learn. They're not double exponential, sharp, followed by a slower ramp. Instead, they learn kind of slowly and consistently, but they do get to symmetry, and they have an after effect that slowly decays. Now, if you look at their perceptual change after learning, like I've shown you before, what you see is that adults recalibrate how fast they need to make that fast leg go, but the children don't do it as much in this 6 to 8-year-old range. Now, what's interesting and what is not reflected here is if you look at individual children, this average sort of reflects that some recalibrate really normally or adult-like, and some don't at all. And so there's a step function transition going on here. It's more all or none. It's not as graded as one might expect. So this tells us that what's coming online in this, honestly, it's not really mature probably till age 12, is this ability to learn a locomotor pattern like adults, and particularly the part of it that makes the initial learning fast and recalibrates perception, which we think might be cerebellum dependent. Now, important for you, and I'll only tell you about one study about this phenomenon, but I think it's really critical for anybody interested in rehabilitation, is whether what you do on any device transfers at all to natural movement. And everybody here probably knows that that's a big issue. So we did a study where we looked to see how well children and adults, healthy children and adults, typically developing children, transfer to overground walking, that's just walking on the ground, from learning on the treadmill. So we tested them walking on the ground, we tested them walking at baseline on the treadmill, split belt learning, then we took them off the treadmill, plunked them down on the ground, and looked to see if they express the after effect that we expect to see, which is our marker of learning. And then we took them back to the treadmill. Again, another large-ish study, 42 kids of different ages and adults. So what I'm going to show you on the next slide is how much they learned in the form of a catch trial. This is in the motor domain, and then I'm going to show you how much they transferred. So the learning is here on the top. And so this is the amount learned, the asymmetry learned. 0.2 is good. That's nice, robust learning. And what you see is we got everybody, except for the adults, looked like they learned a touch more, but everybody learned about the same amount. That's important on the treadmill. But look what happens when you go to overground walking. The little kids transfer a lot. In other words, you take them off the treadmill, and they keep doing what they were doing on the treadmill, which means that if we train them on the treadmill and we put them overground, they will generalize. And that means the training on the treadmill might actually impact natural walking. But look what happens to the older folks. The transfer is not very good. So this is not just a group phenomenon. If you look at individual subjects, you see sort of it's not a perfect line, so it looks a little bit exponential in terms of the way this transfer works. So we now know that children can learn. Younger children learn slower. They're missing maybe part of the neurobiological process of this learning, but they transfer really well to overground walking, or at least better. So that's cool. The last thing I'm going to show you in this healthy part of the talk is what happens if you try to build upon what you're learning. And so this was another rather large study that Kristen Musselman, who's in the audience, did in a really short period of time. It was remarkable. The efficiency of your advisor is shocking. And after being out on maternity leave for a year, which in the US we were like, a year? Really? Which I think is great, but it was still shocking. Because she arrived like this. Anyhow, she decided to see if there was, how well children could benefit from multiple training sessions. And we did something that is in the motor control literature that we think is very interesting, and I'm going to explain it to you to show you another phenomenon in children that is not mature. So here's the task. You come in on day one, and this condition I'm going to call relearning. You walk at baseline, which is tied belts, and then you do 3 5-minute split belt learning sessions. Okay, so you learn the new pattern. You go home, you come in the next day, and because it doesn't generalize or transfer fully, you still have some of that stored calibration. You get on the treadmill and you actually limp when we turn on the tied belts. So you've got a stored, we wash it out, and then we say, can you learn any faster on day 2 after we get rid of the whole thing? Do you build upon what you've learned? The other thing we tested is whether you retain. So it's a bit different. You have a baseline period, you adapt or learn on the split belts, you go home, and then we come in the next day and say, is any of it left? So that's more like a memory, and this is more like washing out a memory and seeing if being exposed to it before helps you to learn faster. Does that make sense to everybody? Good. Okay. So here's what this is actually one of the children in this study. So this is actual individual data. This again is our step length asymmetry measure. 0 is symmetric. So here's baseline, learning our 3 bouts, get all the way back up to symmetry, go home. And this is an older kid, so what you see is they have this giant after effect. They go home, but they've got this stored thing, and they come back in, and they have to wash it all out. And then when they adapt a second time or learn a second time, they do better than they did here. You see how much faster this is? It starts higher, and it's faster. If they do the same thing where they just have a baseline, they learn, they go home, and they come back in, there's a little perturbation. So this is just re-exposing them to the split belt, but they hold on to it. So they retain really well. Now let me show you this across groups. Now, we're going to start from the bottom. This is the relearning group. So day 1 is in purple here for the older kids. They adapt. We wash them all out, and then on day 2, we readapt them after we wash them out. So there's a real benefit from day 1 to day 2. You see this gap? So not only do they retain it, but if we wash it away, they can relearn it faster. They have some memory that's stored. But look at these guys. again, up until 6 to 8, 9 to 11, if you wash out their walking, their adaptation the next day, and you do it again, they act like they've never seen it before, which is really surprising, don't you think, Kristen? Yep. Now, this is what's, so that's that. Now, if you look at how well they retain it, again, day 1, and instead of washing it out on day 2, we just look to see, hey, can you remember doing it before? And everybody can remember doing it before, but if you wash it out and try to start again, the little kids lose it. So these experiments, what I want you to get from this is, first of all, below age 12, adaptation for locomotion, this kind of cerebellum-dependent learning is immature. It's slower. It doesn't seem to recalibrate everything that we might expect, perception and action. And that tells us that the entire nervous system that does this job is not online yet. Little kids, though, transfer to natural walking better. And that might be interesting, and you might be able to take advantage of that. And then little kids, if you wash them out completely, they show less benefit in day-to-day learning. So these might seem like phenomena that may or may not relate to therapy, but I think that they do, and I'm going to show you now what happens when you have a brain lesion. But from a basic point of view, it's really interesting to understand why their learning is immature to begin with, because that's something that in a really little kid you'd rather not tell them how to move, because they'll only do so much. It's nice to be able to put them in a situation that just automatically induces the learning, like this treadmill. And so why is it immature? Well, it could be because we know primary sensory and motor cortical areas develop a little early, but they're still delayed. Cerebellar peak volumes, though, are really delayed. And so we think that this might reflect the cerebellar system coming online fully. It's a hypothesis. I don't have the exact data to prove it, but if you take all of the pieces, that's what fits, in part. Okay, now let's talk a little bit about patients. This is another animation, and this is actually an adult who's had a stroke. They've had a stroke, in this case it would be on the right side of the brain, the right cerebrum, causing an impairment in their left blue leg. And so what can we do for these people? Well, as you know, there are lots of therapy trials involving body weight support treadmill training and making people walk fast and doing functional electrical stimulation, and they work to some extent. But in rehab, it's never one size fits all, and so we'd like to understand something about the neural substrates involved in helping these people to learn more. And so what we found through a series of studies that I'm going to summarize now is, first of all, I told you that at least the cerebellum is an important part of the circuit for this learning. And what I haven't told you totally yet is that adults and kids with damage to the cerebrum actually do adapt very robustly. So the guy with the limp, I can put him on the treadmill and he will learn and show an after effect that can affect his gait or her gait. Now the cool thing, which is that if you have a cerebral lesion, you transfer really well to overground walking. And we published a couple of studies on this transfer. So what I showed you before was that little kids transfer well, adults transfer lousy. Well, if you're an adult and you've had a stroke, you transfer well. And if you're a kid and you've had a cerebral lesion, you also transfer well. So that tells you that maybe you need the cerebral inputs to be able to make your learning context specific. So maybe this is actually a deficit. Does that make sense to everybody? So you don't want to store the same caliber. You don't want to always use the same. Let me give you another example. You know how when you drive a car and it's your car and then you go drive a rental car and it feels really weird? because the amount of force you have to use to turn the steering wheel is different, the amount of force on the brake and on the accelerator, it's all different. Not just things are in different places, but the dynamics are different. And then when you go back to your own car, how does it feel? Does it feel weird? It usually feels a little bit weird if you drove the rental car enough. That's because you're recalibrating the same motor actions. Now, if you drive two cars enough, you can flip back and forth and you don't mess up one calibration with the other. I think that what's happening here is that they're using the same calibration for everything, and that's why it's transferring to overground walking, whereas healthy individuals might be able to form specific memories. Okay, that's my side. And then I'm going to show you that if we use this as a starting point, we can actually improve long-term training. Okay, so some of you may know this, and I think this is true in lots of domains, but when we apply this to rehabilitation, it turns out that we have to make a person's limp worse in order to help them learn how to correct it. So you might imagine that if I put you on the treadmill and I can change the speeds of the belts, I could correct your limp for you. I could dial in the right speed and help you. But it turns out you always adapt back to your own baseline. And so that ends up making you worse once we take you off the treadmill. And so instead, what we do is what we've done here. This is the MRI from this individual. These are the step lengths. Instead of symmetry, I've broken them out. Here's the long step and the short step. The non-paretic leg takes a longer step. The good leg takes a longer step. The bad leg takes a shorter step. When the belts are going slow at the same speed, whoops, or fast at the same speed. So then we go back to slow. We try to teach people this new pattern, but what we do is we make their limp worse so they have to learn how to correct it. And so the nervous system automatically starts to do that. And that gives them, when we go back to tied slow, an after effect that meets this step asymmetry in the middle. In more recent times, we've actually compared this to just asking people, please try to walk as even as you can. And the way that you do it here is, I'm going to say, first of all, more complete, and it's better because they start taking a longer step on the short step leg, whereas if I ask you to take even steps, everybody just shortens their longer step. That may not make sense to everybody, but I'll show you something in a minute that'll help. Okay, so you have to exacerbate the limp. So what that means, you have to put the leg that takes the smaller step on the fast belt, and you take the leg that takes the bigger step or the longer step on the slow belt. Okay. Okay, so I've had the opportunity to study, and I don't know how many kids you guys have seen here with hemispherectomy. So this is a procedure done for intractable seizure disorders. For those of you who haven't seen some of these kids, instead of removing the focus of a seizure, they remove a whole hemisphere of the brain. And so that's what's shown here. This is this little girl's MRI, and it's shown from the back of the head. So here's the cerebellum, which is nice and intact. Here's one hemisphere, and then here is CSF. So this is the whole. They've removed the whole hemisphere. This little girl was one of many kids that we were able to train, and I'm going to show you what we did for her. This is a single session, and I'm going to highlight the fact that we can get them really good in a single session, but it's going to wash away because the nature of this learning is the time scale is slow. You have to do multiple sessions and some other things to make it stick. But let's look at how she walks. Her bad leg, her paretic leg is back here. You can see her AFO. Here's her good leg. And sometimes you'll see this woman here stick her hand between her legs so she doesn't cross the belts because the belts are going to be moving at different speeds and it spins you around. Okay, here we go. So she vaults on the leg closer to you to try to get that bad leg through. And she takes uneven steps. And I walk in front of the camera. And so then we actually make it harder for her. So the leg closer to us is now the fast leg. And you might see she's kind of stumbling a little bit more. That's a 2-to-1 split. It's a slow speed, so it's a little harder to see. So we give her that training for 10 minutes, and then we go back to the tied belts, which is exactly like this situation, but now 10 minutes later after doing this. And this is what she can do. So what we've done is we've used subcortical mechanisms to actually help her to learn this new stepping pattern in time and space. Now, that's on the treadmill. So you should be happy that she has the capacity to do this, but you should be unhappy with me that I'm showing you this on a treadmill, not over ground walking, which is what you really care about. And so it turns out that when we take them over ground, they can do this as well, but we have to reinforce it in order to get it to stick. So it's not just adapting it. We also have to give them, and we're learning that we're going to have to give them even more temporally specific feedback. But we've been able to do this in kids and adults, and I'll show you some data. But it's always fun to see. This is this kid when she's 13. I can't definitively say that our intervention did this because natural recovery, of course, is robust. But she walks pretty well. You can see that her hand there never really recovers, and that's because the hand representation, it's one of the things to think about when we think about legs versus arm. The hand representation is special to motor cortex, and there's not another really good hand representation in the brain. Whereas the leg is in motor cortex, but there are also all these subcortical mechanisms to help control the leg in the context of walking. So you have a little more to work with there. Anyhow, I just like seeing her. She's walking in Crocs, and she's walking pretty well, which is awesome. Okay. So oftentimes we only had 2 weeks, and I have to say, my hat's off to all of you who do these training studies, because they're hard, and children are moving targets, and getting the right controls is incredibly challenging. And so single case designs, I think, are going to be really important. This is a kid that had another disease called hemimegalencephaly, and hemimegalencephalopathy. And so this kid took uneven steps. Green is one step and blue is the other step, and you can see these two bars aren't even. Training day 1 post-surgery. We got to see these kids really quickly after their surgery. Here's day 11. This isn't terrible, but it's asymmetric. And then we split belt trained this person, and then they got longer with both steps. But, you know, they weren't that asymmetric to begin with, so what do you make of that? Here's another one. This is somebody with Rasmussen's encephalopathy, which is a more aggressive seizure disorder. And you can see on day 1, highly variable hemiparetic step, really short non-paretic step, really had trouble walking. This is training day 12. So symmetric, and again, meeting in the middle. It's not like they just made this step really, really short, which is what I was referring to earlier, which is what you don't want. And if you tell somebody, take even steps, that's what they do. They shorten that step. And 7 months after surgery, there's a little bit of, it was certainly better than here, but, you know, it's not perfect. This is a kid who had a stroke, who had a, robust asymmetry, and we just did tied belt training, just regular old treadmill training, and we didn't really change the asymmetry. By the way, these are all measurements taken over ground. These are not treadmill measurements. Adults are easier, unfortunately, because they're not a moving target. Children are developing and they have a lesion. And that's why understanding development of these learning mechanisms is so important. Adults, we've done the study, and a month of training does have long-term effects. And I'm not the only person who's shown that. Some of your colleagues, Sophie Nadeau, has shown this. This is baseline. Here's one step, non-paretic, paretic. In this case, the paretic step is longer in this group. What that means is the non-paretic step is shorter because they have trouble standing on the paretic leg. In any case, asymmetric, asymmetric. Post 4 months training, we get them better, and luckily we make them having longer steps, not perfect. We stopped the training here. A month later, they held on to it. 3 months later, they're slowly even getting better. So something else is happening. It is not just adaptation. It cannot just be that. But it did help kickstart them in the right direction, and that's my main point. Now, what we found since then is that if you walk slower than 0.4 meters per second, which is really slow, this doesn't help you. And I think it's because you're more like standing, stepping, standing, stepping. It's not necessarily the same fluid pattern of walking. That's a hypothesis. Okay, so I'm almost done. And I'm going to summarize what I've told you, and I'm going to tell you just a couple things that I think are interesting, and then I'll let you go, because I think it's a little warm, and, but I appreciate the attention. So what I've told you is that we've studied one piece of a learning process, adaptive learning, in one movement type, walking. And you can see the great pains we've taken to try to characterize this, understand how it develops, understand what brain circuits you might need for it, understand how it transfers to natural movements. And what we found is it's immature below age 12. It relies on cerebellar integrity. It generalizes differently depending on if you're young or if you've had a brain lesion. So you have to study what you want to study. You can't take adults and say, oh, kids will look just like that. You can't do that. You guys know this, I know. But you also can't say, oh, the normal behavior will absolutely play out in the people that have different kinds of damage to the nervous system. We're really interested in why we're seeing the things we're seeing, associated, of course, with brain development. Age 12 is getting close to puberty. We know other learning mechanisms are online during this. So we're exploring these things in different ways. Adaptation can be used for rehabilitation of walking. We've shown you split belt. There are other ways of adapting people. Here we had to augment their asymmetry. We had to give them a perturbation that makes them worse so they learn how to correct that abnormality. I don't know. I have not done the definitive study in kids. We've done a couple studies in adults, as have others. I don't know in my Hemi kids how much is training and how much is recovery. So I'm giving you all the caveats to be absolutely fair and transparent. I don't know what the dose should be. This is rehab, and everybody here understands this. These are hard studies to do. But they're incredibly important because no matter what drug you give somebody, no matter if you give them stem cells or gene therapy or robotics or prosthetics, people have to learn how to control their body to be effective. And the more we know about that learning process, the more we're going to be able to do. Now, in my last 3 slides, I'm just going to tell you a few things, which is I mentioned we've been studying this, adaptation-based learning, which we think comes online in this phase of childhood, at least fully. We're starting to study really explicit learning, telling people exactly how to move and seeing if they can do it. It doesn't work that great in kids so far. Yeah, surprise. It does work better in adults, and we're taking high dimensional feedback and we're reducing it to single dimensions to give them continuous online feedback about walking. And we're able to make some changes. Reinforcement learning, people think, is actually driving a different circuit. So adaptation is driving a cerebellum-dependent circuit, a circuit that involves the cerebellum. This probably involves the basal ganglia. And we have been able to take people with cerebellar damage and use this kind of mechanism to help them to learn better than we can with adaptation. So this is pretty interesting. Of course, a purely cortical type of learning is use-dependent learning. If you do it enough, again and again, you get a Hebbian-like mechanism that can kick in and reinforce that. How to layer, and these are just some of the things that are happening all the time in your brain. So how to layer these things, how to try to force the system to use one more than the other is one of the things that we're very interested in. And when do they start really being used? When are they mature? So earlier it was mentioned, and I am a believer in this, that the earliest rehabilitation training is going to be the best. So this is this classic article showing synapse formation. These are neurons. These are pyramidal cells in the cerebral cortex. This is at 36 weeks gestation, newborn, 3 months, 6 months, and 2 years. What you notice is that the neurons, of course, get bigger, but they get much more interconnected. So as you hit age 2, 2 is when you have the densest connectivity in your cerebral cortex. And the estimate is that 700 of these contacts between neurons is forming every second, which is wild. And now, I mean, there are techniques to image this now using 2-photon microscopy. So that's amazing, but by 2 years old, you've got actually more connections than you really want. And what happens, so at 2, twice the number as an adult. That doesn't mean your 2-year-old is twice as smart as you, so don't misinterpret. But you start to prune, and this is the really critical thing. You're pruning age 2 years plus. This shows out to 6 years. And the connections that you're using, this is the Hebbian kind of mechanism, are more likely to stick. So if we're not using the things we want in that critical period, it's going to be harder to get them back. So the hard connections are being formed in this very early time period. So my feeling is that if we can go low, we're going to be able to go early, we're going to be able to do a better job. And we don't know how much the dose should be, but wrapping all the way back, my last slide, to our children better learners in development. As I mentioned, either good or bad experience is important, but the dose. Well, when you're learning to walk, I love Karen Adolph's work. I'm sure some of you know of her. Children are taking about 2,400 steps per hour, which is about the length of a football field when they're learning to walk. So, and they don't mind falling. I mean, they do eventually, but they get back up. They do it again and again and again and again. And so the kind of dose, first of all, saying children learn better than adults. Well, if adults practiced as much as children did, it would be a fairer comparison. And children also are in this critical period as they're learning to walk. And if they're getting good experience, those circuits stay. And if they're getting bad experience, guess what? Those circuits stay. So that's why I think it's so important to go young. Okay, I will wrap up now. These are many of the not all of them, unfortunately. I love students and postdocs. That's training is a huge part of my life, and so these are some of the trainees. I want to highlight 5 Canadians who've worked in the lab over the years. Hannah normally does not wear a Star Trek uniform, but she just got tenure at the University of Indiana and had a bet with her friend and said, well, if I get tenure, I can wear anything I want to faculty meeting. And this is what she wore. And so she sent me a picture, which made me extremely happy. The other person who did a lot of the pediatric work is Jen Keller and Helsi. So Helsi is the other border country. She's from Mexico. And so you can see that basically what used to be NAFTA worked on this project. I don't know what it is now. I shouldn't go there. And of course, we are so thankful for having funding to be able to do these things. So thanks for coming to the talk and listening to this story, and I'm happy if you have any questions either here or after the talk.
I just want to thank, again, Dr. Amy Bastian for joining us this morning and inspiring us with such an amazing talk. Thank you very much. Yeah, thank you. Thank you. And thank you all for joining us this afternoon.